Welcome to the ICAR-National Rice Research Institute, Cuttack. Established way back in 1946, the institute has been conducting experiments in its experimental farm for various aspects of rice on productivity enhancement, nutrition, climate change, soil health, resource conservation, biodiversity, farmers' profitability and others. This institute has well laid out farm having 22 blocks, 776 plots covering 160 hectares of land with irrigation and drainage facilities. The important field research activities include hybrid rice production, new generation rice, characterization of germplasm, biofortification, generation advancement, breeder and truthful label seed production, AI therapy trial, quality rice production, advancement and multiplication of elite breeding lines for biotic and abiotic stress, integrated farming system, long-term fertilizer experiment, natural and organic farming, and other experiments. The institute is having spread breeding facility, submergence tank, rain-out shelter, open-top chamber, off-season nursery facilities, aerobic rice research block, a Oryza garden with 24 taxonomically authenticated species of genus Oryza. Let's have a glance of our institute farm.
The crop cafeteria plays a pivotal role in fostering collaboration and knowledge sharing. It is located in the A blocks of the NRRI field, which provides a single window platform for various rice stakeholders like any profit organizations and companies or a not-for-profit organization to gain the first-hand information on the advanced varietal technology of the institute. Any collaborators can benefit from the technologies of the institute by taking up membership for the technology sharing. Hybrid rice technology is transforming agriculture, enhancing yields, disease resistance, and quality. At National Rice Research Institute, we focus on developing rain-fed shallow lowland rice hybrids. Our portfolio includes 6 hybrids: Ajay, Rajlakshmi, CR Dhan 701, CR Dhan 702, CR Dhan 703, and CR Dhan 704 suitable for different ecosystems. We maintain diverse parental lines for crossbreeding and enhancing them for salinity, submergence, and herbicide tolerance. Advanced breeding populations for these traits can be seen on our hybrid rice production plots. We also produce breeder seeds for these hybrids to support the national seed supply network. Hybrid rice technology promises a more sustainable and prosperous agricultural future. Facing climate change vulnerabilities, India emphasizes biodiversity conservation. The National Rice Research Institute preserves 30,000 plus rice germplasms in the gene bank, vital for better rice varieties. The germplasm panel development project characterized donors to boost the national rice breeding program. Currently, 2,500 germplasms, including donors for biotic and abiotic stress tolerance, grain yield, nutrient use efficiency, grain nutrition quality, indigenous varieties and aromatic rice germplasms are raised for purification. The selected panel will undergo phenotypic evaluation and genotyping to strengthen breeding. This diverse germplasm supports global food security and aids public and private crop improvement agencies. To address protein energy malnutrition, integrating nutritionally enriched foods like biofortified rice into daily consumption can be a potential solution. National Rice Research Institute offers 4 biofortified rice varieties such as CR Dhan 310 with high protein, CR Dhan 311 with high protein and moderately high zinc, CR Dhan 315 with high zinc and CR Dhan 411 with high protein in Swarna background. Honorable Prime Minister of India dedicated CR Dhan 315 to the nation on the 75th anniversary of the Food and Agricultural Organization, marking a significant step in addressing alleviation of malnutrition in India. All India Coordinated Research Project on Rice is one of the major activities of NRRI Cuttack. Being the coordinating center for the rain-fed ecology, NRRI is engaged in coordination, monitoring and reporting about the ICAR-NRRI trials to the lead center. NRRI also undertake agronomic, entomological, pathological and plant breeding trials for the irrigated rice ecologies. Such trials under ICAR-NRRI helps in identification of most stable, high-yielding and superior genotypes for different agroclimatic conditions and possessing required level of resistance to the targeted insect pests and diseases. The crop cafeteria plays a pivotal role in fostering collaboration and knowledge sharing. It is located in the B block of the NRRI field, which provides a single window platform for various rice stakeholders like any profit organizations and companies or a not-for-profit organization to gain the first-hand information on the advanced varietal technology of the institute. Any collaborators can benefit from the technologies of the institute by taking up membership for the technology sharing.
Aroma enhances culinary delight, and India possesses numerous aromatic rice varieties such as Kala Namak, Kala Zira, and Pimpudibasa. ICAR National Rice Research Institute, Cuttack, genetically enhances non-basmati aromatic rice for improved yield and stress tolerance. 11 non-basmati aromatic rice varieties like Noakala Zira and Siar Sugandh Dhan 911 offer yields from 2.5 to 5 tons per hectare. The focus in 2023 is on Gobind Bhog rice, holding a GI tag in 8 West Bengal districts, and antioxidant-rich black rice, with promising Chakao derivatives displaying 100% grain filling under Cuttack's climate, set for testing in ICAR-NRRI.
Climate change increases abiotic stress, demanding climate-resilient crops. Using double haploid technique (NRRI), Cuttack expedites climate-resilient variety development process. It stabilizes breeding populations in 1 to 2 years, fast-tracking cultivar release. Using this technique, the institute has released 5 high-yielding rice varieties, including nutritionally rich CR Dhan 324 and CR Dhan 911. Open-top chambers are weather-simulating structures which are used to elevate air and soil temperature in the warming experiments. NRRI has this facility for conducting different experiments at elevated CO2 concentrations. The nucleus seeds of the varieties are produced to generate a sufficient quantity of seed for the next generation of seed multiplication. The bulk nucleus seed is used to produce the breeder seed. TL seed is produced to meet farmers' requirements for rice seeds. The institute is producing TL seed of more than 50 rice varieties. NRRI variety, Hanseswari, is highly tolerant to pHs. 12 genotypes were observed to be resistant to pHs. At NRRI, we screen large numbers of genotypes by exposing them to a light cut to the tune of 25 to 50%. At NRRI, we evaluate tolerance of rice genotypes to high temperature stress under field conditions by adopting the staggered sowing method. Diseases caused by viruses pose a threat to rice cultivation in a changing global climatic condition. Monitoring of viral diseases in rice germplasms is carried out based on phenotypic symptoms and vector population. The breeder seed indent is being received from DAC, different states, private organizations, seed growers and other agencies. NRRI produces breeder seeds of 70 varieties. long-term fertilizer experiments are invaluable in assessing changes in crop productivity, soil characteristics, nutrient imbalances, and soil health. NRRI Cuttack houses a long-term fertilizer experiment on rice cropping since 1969, involving 10 treatment combinations of N, P, K and FYM. The experiment helped in evaluating soil properties, crop productivity, and their responses to different nutrient inputs for responsible fertilizer use and minimizing environmental degradation. The organic and natural farming addresses the negative impacts of conventional chemical farming, offering insights into profitable, eco-friendly alternatives. These experiments were initiated in 2022-23 in our farm to evaluate nutrient management, pest control, and farming practices under various cropping systems. At our N block, these experiments are conducted. Such experiments are crucial in improving farming practices and livelihoods while minimizing environmental casualties. Annually, new rice varieties are released by multiple institutions and universities involved in rice improvement. To protect these, India enacted the Protection of Plant Varieties and Farmers Act (PTV) and FRA in 2001. The National Rice Research Institute in Cuttack is a dust testing center assessing candidate rice varieties and hybrids based on 64 morphological characteristics. In 2023, first-year testing covers 13 candidate varieties, 12 hybrids, 1 farmer's variety, and 96 notified varieties, following a randomized complete block design with 3 replications. The Institute's dust testing plays a vital role in providing, characterizing, and protecting new rice varieties through PTV and FRA. The Odisha Reference Garden has been established through collaborative efforts of NRRI and NBPGR Base Center, Cuttack. Currently, there are 24 taxonomically authenticated species available in the genus Odisha. Only 2 of those, Odisha sativa and Odisha glaberrima, are cultivated and the rest are wild. The wild Odisha species serve as reservoirs of novel genes and alleles required to face the challenges arising from climate change besides the ever-evolving pathogens and insect pests. The Odisha Reference Garden of NRRI has 23 species of genus Odisha representing 4 major species complexes and 2 outgroups. Besides use in rice genetic improvement, the reference garden will serve as live taxonomic reference of genus Odisha for students and academicians. ICAR National Rice Research Institute Cuttack prioritize the importance of quality seeds for crop production. As a premier rice research institute, we focus on providing top quality seeds to farmers. During Kharif 2023, we've planted 116 nucleus seed varieties in 8 plots in our T block to produce high-quality breeder seeds, supporting the dissemination of varietal technologies. Our C, D, E, F, G, and J blocks, spanning 45 to 50 acres, are dedicated to breeder seed production, encompassing various ecologies and transplanting durations. In Kharif 2023, we are planting 61 varieties for breeder seed production. Our mission is to ensure food security and improve the livelihoods of farmers by offering genetically pure breeder seeds. Intermittent floods pose a threat to global rice crops, impacting food security and farmer income. The sub-1 genes discovery has offered solutions to submergence issues. At the C block of NRRI farm, our submergence tank tests rice germplasms for submergence tolerance. We assess sub-1 containing rice varieties against complete submergence, examining potential yield loss, morphophysiological traits, and greenhouse gas emissions. We are also evaluating 100 germplasm accessions for flash and stagnant flooding tolerance to identify resilient cultivars for different growth stages, strengthening climate resilience and protecting farmers. The looming irrigation water crisis is seriously affecting the rice cultivation, and hence there is a need to grow more rice with less water. Adopting aerobic rice coupled with alternate wetting and drying method conserves around 30 to 50% irrigation water and also mitigates methane emission up to 30%. Despite a 10 to 20% yield penalty, improved rice varieties suitable for aerobic condition offset this. NRRI Cuttack has released 12 high-yielding aerobic rice varieties like ICAR-Dhan 200 and ICAR-Dhan 201, which are resilient to abiotic and biotic stresses. The research trials at Y block of experimental farm of institute focuses on development of rice varieties suitable for aerobic conditions.